My name is Michael Johnston. I'm a third-year graduate student in Todd Neistel's lab at the University of California, San Francisco. In the Neistel lab, we are studying the Drosophila ovary. So if you don't know what a Drosophila is, that's a fruit fly. And it's a really simple tissue to look at when you want to look at adult stem cells. So these aren't the embryonic stem cells that everyone sort of gets up in arms about in the media. These are adult stem cells that are used to replace tissues that have really high rates of turnover, like your skin, your intestines, even breast tissue. And in case those tissues sound familiar, that's because those are tissues that frequently get cancer as we get older. I grew up in Swan River, Manitoba. It's a town of about 4,000 people. I'd say, 5 hours away from the nearest Walmart in Canada. My dad and mom both grew up in farming families in Western Canada. My dad's an accountant, my mom's an English teacher. I think it was really a great chemistry teacher that I had in high school that really got me sort of inspired to questioning things in a little bit more detail than I had before. From working with my dad around farming stuff, I'd always seen like NPK ratings on fertilizer and never really known what they meant. But it was when I got to chemistry class, like, oh, NPK, those are elements. These are things that are important for the soil, for the plants that we're growing. And I sort of got interested in the chemistry behind all of the farming that I had grown up in around my entire life.
Hi, my name is Michael Johnston, and welcome to the Neistel lab. We've got 2 bays for our lab. This is Rebecca hard at work.
We are working with fruit flies, so we've got a whole array of great genetic tools that we can use. There are many mutations in different genes that we find very important when we mutate them. We're trying to study how the stem cell populations are maintained and regulated by different signals in the body, with the hopes that if we understand how they function normally, we'll be able better to get them back on track if they ever go awry in tumors of these tissues. Just naturally, in the course of studying these important regulators, we stumble into things that are causing tumors and do cause very drastic effects in the tissue. So in that sense, it's also studying the tumorigenic properties, the cancer-causing mutations in these adult stem cells. I guess it's hard to boil that down to an average typical day, because I do have a very varied schedule in terms of the techniques I'm using, the things that I like to do. But that's one of the things that really draws me into the job. I feel like I wouldn't be able to be like my dad. I couldn't be an accountant. I couldn't look at the same sort of tax forms every day and still like wake up excited to go to work. Today I'm working on, digesting some DNA to make some probes so that I can look at what genes are going to be made in certain cell types. Another day I might be, trying to sequence the genotype of a fly to figure out what might be wrong with it. And I think it's that variety that really keeps me drawn in every day. I think that anytime you work on a team, it depends a lot on your team, and the Neistel lab's got a great team. We've got a whole host of people from very young scientists who haven't even entered grad school to people who are postdocs who've got extensive experience both in stem cells, in how the germline develops, and working with fruit flies. So one thing that is common amongst most of the scientists that I know is that we love problem solving. So the day that your experiment doesn't work and you're trying to figure out what it is, as long as you've got all the appropriate controls laid out, you can sit down and try to analyze it with the help of the other scientists who are like, okay, let's try and rebuild this puzzle and figure out what actually went on. The goal shouldn't be to try to determine exactly where you want to end up at the end of the day. I think that there's enough different flavors of science you can work on that look at what is tractable and interesting to you right now and just do as much of that as you can. I chose chemistry because I thought chemistry was really cool, and eventually I ended up transitioning out of chemistry because the techniques that I was using were also applicable to stem cell biology. So I don't think the goal should be to look way down the line and try and decide exactly where you need to end up and meet every checklist on the way there. I think that if you find the avenue of science, math, technology that really excites you, that engages you, and that you're good at and want to keep doing today, do it tomorrow, keep working at it, and eventually you will find an excellent niche to fill in the scientific community.